All right, great. Welcome, everybody. It's a real pleasure to have Hector Carrada Bravo from Gentech, and I guess Jay is going to talk as well. So we have a dual set of presenters. As Jacqueline said, I have known Hector for a long time. He did his PhD in Wisconsin with Grace Waba, who is often known by the nickname of Amazing Grace. Then he went and did a postdoc with Rafa for a while. Is that right, Hector? I think that's what I'm remembering.
All right, yeah.
At Hopkins, and then he took a faculty position at the University of Maryland. He has worked on a number of really interesting and important problems in computational biology, often with a strong emphasis on really great statistics and really great visualization. And today, he and Jay are here to talk to us about some work they've been doing at Gentech around the creation of a SingleCellHub. And without further ado, Hector, I'll turn it over to you guys.
Awesome.
Is it useful for me to watch the chat for questions? How do you guys want to handle that?
Yeah, absolutely. Just questions along the way would be great.
Okay.
All right. So thank you very much for the introduction, Robert. So Jay and I were both at the University of Maryland, actually, the Center for Computational, Bioinformatics and Computational Biology. I've been working on, statistical and software systems for genomics data analysis, interactive genomics data analysis for quite a bit. And since we landed in Genentech about 2 years ago, to a little bit over 2 years ago, we started this project on the SingleCellHub and thought we'd talk about it today. So I serve as the kind of scientific lead of the project. Jay is the engineering lead, and we'll present both sides of that, of those aspects of the project as we go along. So if you'll allow me, all right. So disclaimer, don't take anything I say as a medical advice. All right, so I'll start at the beginning. So we're working on the SingleCellHub, but I thought it would be useful to put this work in a little bit of context. And with that, I'll start with Sidney Brenner. So Sidney Brenner won the Nobel Prize in 2002 in physiology and medicine, and won it for really kind of fundamental genetics research on apoptosis and animal development, right? The most kind of reductionist genetics research you can imagine. Knock out a gene, see what it does to, say, a phenotype, right? So knock out a gene and see how that affects development and ultimately the final phenotype of C. elegans, which he established as a model organism. Little note of history, that's Sidney and Sidney Brenner in the Asilomar conference. It's an important conference for us in biotech. But the important thing is, when, as a Nobel Prize winner, he gets to give a lecture. The lecture he gave in 2002 is a reflection of his work in, as I mentioned, pretty reductionist genetics research. But along the way, he also provides a perspective of work he thinks would be useful to embark upon in the 20 years from that point, 2002. And he brings up 2 projects, the first of which is what he called the cell map, which is exactly that, mapping gene expression at cell type levels across the organism, and makes a remark about how at 2002, so genocentric modern biology has become that we have forgotten that the real units of function and structure in an organism are cells and not genes, which I think it's a useful thing to think about, and really a lot of the motivation behind the work that we're doing. Now, technologically, it took some time for that to start happening. It's about 15 years later, in 2017, that the beginnings of the Human Cell Atlas became more of a reality, or at least something to strive for. And in the 5 years since then, as you are probably very aware, lots and lots of single cell data across a number of systems or organisms has been generated. And that's useful because at least us in drug discovery, drug development, we understand, or at least we think, that our ability to discover efficacious and safe therapeutic targets just gets better the more we understand cellular state and how those cellular states are organized in homeostatic and diseased tissue. So a couple of examples in neuroscience. If we want to understand the effect of neurotoxicity in neurons, it would be useful to understand what neuronal states render them vulnerable, what neuronal states render them resilient or resistant to neurotoxicity. In the tumor microenvironment, we would like to understand how the cells in the immune system and their interplay with cells in the tumor works, so that we can start thinking about immunotherapies for tumors. And in infectious disease, the more we understand the specificity of cell types that make part of the epithelial barrier and their interaction with pathogenic with pathogens, the better we have a chance of understanding inflammation and other processes in infectious disease. So, this is not new. I think a lot of the experiments, well, definitely a lot of the experiments with Gentech has a pretty strong component of single cell data generation. But for us, specifically, a question that we're thinking through is how do we use large scale genomic data sets or data collections to help understand cell states and their organization. And for today, our focus will be on transcriptomic cell state as we understand it from single cell, or as we measure it from single cell RNA-seq. And I'd like to put some of this work in a particular context. I like to think of the way we can use these kind of large data collections as a progression, where we first observe, then recall, then synthesize, and ultimately predict. And what I mean by that is we first, if we want to understand cell state and their organization, well, the first thing to do, seems reasonable, is to observe cell states and organization. So we've spent some time collecting, curating, and harmonizing data sets, and we'll talk a little bit about that. More importantly, I think the next stage where we go from babies to toddlers is really, can we find those data sets in useful, meaningful ways? And the better that we are at representing cellular state in tissue and disease context, the better we are at finding those appropriate data sets. And really, I think it's also useful to think about how do we move from data set as the unit that we care about to cell state as the unit that we care about. And how do we query and find cell states in context as an important activity? And we'll talk a little bit more about that. Ultimately, what we'd like to use this data abstractly and at the highest level is to be able to start thinking about prediction, right? So predicting the effect of either chemical or genetic perturbations on cellular state in specific tissues and disease contexts. And while I don't think with this kind of observational data that we'll be discussing today, we'll get to we'll make a lot of headway in the prediction problem. We'll make some headway, but not a lot of headway without direct measurement of perturbation effects. There is going to be a limitation to how much direct observation of perturbation data we can generate. So therefore, using this kind of large-scale observational data will allow us to put those perturbations in context and start maybe thinking about extending through predictive methods what we observe in direct perturbation experiments. But we won't talk very much about this prediction side of things. We'll concentrate on the observational regime and talk about the work that we've done for collection, query, and representation of cell states in this project. So we've all of these things are great as a exercise. Academic exercise, or at least intellectual exercise, but it's also the underpinning of systems. So we stand up applications for our scientists. Some of this is in different stages of open sourcing and so on. We're still working through exactly how we're going to do that, but for the moment, these are resources that we are building internally. And right now we have, around 130 million or so cells in our data sets, and I'll describe exactly how we're thinking through that. So what we'd like our scientists to, we've seen our scientists doing, and we'd like to see more of, is, we're really thinking of ways in which we can make decisions quickly. So using publicly available data sets to prosecute specific hypotheses is something we're really keen on. So finding the right data sets, annotating them appropriately so we can cast those specific hypotheses, say the expression of a particular gene in a particular context and how that's related to, say, other sample characteristics is an important aspect of that. Additionally, a lot of the single cell data analysis has this flavor of query to reference mapping. A lot of the work that we do in single cell has a component of finding the right sets of cells that we think are meaningful. Sometimes it's by using marker genes to define cell type labels, sometimes it's not. But usually there's some flavor of reference to query work. And so we're using a lot of these systems to first integrate data sets that create specific reference atlases for our therapeutic areas. So for example, our folks working on neuroscience, folks working on immunology, we're helping them kind of create their appropriate reference atlases, and then find those data sets to map and query their specific experiment data. And something that we think would be super helpful as well, especially for in specific contexts, to say immunology and infectious diseases, to think about meta-analysis of cell states across tissue and disease contexts. Once we have data sets across multiple contexts, and understanding what distinguishes, say, specific kinds of T cells in one tissue versus the next, or in one disease context versus the next, and tells us something about the specificity of certain disease processes, would be super helpful to do. So those are the things that we are seeing happen with these kind of collections of data. So the overview for today, we'll talk about kind of three main things. One is, how do we handle data? And Jay is going to talk about that aspect of it, and what we call structural querying. What I mean by that is querying data sets based on its annotated metadata. Then we'll talk about methodology for cell state querying, where we, instead of querying by metadata attributes, we query by properties of the transcriptome itself. And I'll tell you 2 cases of that. And then we'll talk about some of the engineering that we've put together to support that on a real-time cell state querying. And then we'll finish with a discussion of what we're thinking about next. So Jay, we'll talk about our next section.
All right, cool. Thanks, Hector. So one of the things Hector mentioned in the earlier slides is we have a collection of around 130 million cells. So you're probably wondering where all of this data comes from. And the first step in that process is, there's a lot of effort that went into the ingestion and curation of all the publicly available single cell data sets. And these are some of the ingestion procedures that we have in place so that we can curate data externally, publish data from publications. So these are data sets tied to a publication, and it's available through Geo, repositories like Geo or Cell by Gene. Our consortiums like Cell by Gene also publish their data sets with these annotations for cell type disease and tissues. So there's a data curation team that is managed internally that comes up with the standardized procedures on bringing these external data sets in-house. And once you bring data sets from diverse sources or various sources, one of the key problem is how do we harmonize these data sets? So one form of harmonization is this structural data set harmonization. So we make sure if this should be familiar for a lot of folks that are coming from the bioconnector world, where there's the single cell experiment object, that is what we use to consistently represent our data across different sources. So we make sure that all of these data sets that are coming from these sources are stored as single cell experiment objects and MAEs. So these contain assays, which are these gene by cell matrices of expression counts, either counts or normalized counts, row data that contains information about the genes, and then column data that contains metadata about the samples or the cells. And the second form of harmonization is, as I mentioned in the previous, we have row data and col data, which is metadata about the genes or cells. we need some kind of harmonization in place so that we have the data stored consistently across data sets. So one specific type of question is what data, if we have this huge collection of data, can we find myeloid cells? And to be able to find myeloid cells and consistently represent myeloid cells, we take advantage of ontologies. So where the curation process annotates cell type, disease, and tissue labels, and to annotate them, we use standard ontologies like disease ontology and cell type ontology, and the last one I'm forgetting, the Uberon is for tissues. So we use these ontologies to map the terms across data sets for all of the cells. And once we go through a harmonization process for both structure and the metadata, we need a place to store this. And this should be familiar for some of you who heard through Aaron's presentation from last month at the same seminar, where he talked about ArtifactDB and is in the process of being open sourcing it. So ArtifactDB is one of the instances that we use internally, and it's also in the process of being open source. And the basic idea of ArtifactDB is you have data and you have metadata associated with it, along with some permissions on who can access it and who can either read, write, or access the data set itself. So one key part of ArtifactDB is the data itself is considered as a blob, but what really helps in terms of discovering this data is the metadata that is around it. And the ingestion procedures that we define in one of the earlier steps defines that metadata that we annotate each of the data as. And another key part of ArtifactDB is we use a language agnostic storage mechanism to store all the data sets in. So we have interfaces in both Python and R that can access these data sets or parts of the objects itself from this instance in either R, Python. And there's also a work in progress JavaScript library if you want to build web applications around it. And that's one of the systems that's being worked by Sebastian and Aaron, and they're also working with BioConnector to have an instance of that running up. So as I mentioned earlier, so in the ingestion procedures, we define the procedures on how we ingest them and harmonize them. And then the ArtifactDB is how we instantiate these and store them on disk. And that brings us to the next step of the process. And now that we have this collection of data stored, we want to find appropriate and meaningful ways in finding these data sets for our scientists. And that's the recall step of the process. And this is where we have our own separate indexing procedures, and we have a graph database in place to perform these procedures. So I'll walk you through what we do in terms of pre-processing or the indexing. So as I mentioned earlier, ArtifactDB is a collection of data objects along with metadata. So among all of our internal collection of single cell reference data sets, we collect all of these data sets and we harmonize them and we index the fields that we care about. So these are usually cell type, disease, tissue labels, and organism or experiment types. And based on these labels, we build a graph. So for each data set, we have nodes in the graph for each data set that contains its disease, tissue, and cell type labels. Now, some of the diseases are shared because multiple experiments are coming from the same disease or annotated for the same disease, or the cell type labels can be shared as well because when they were curated, they have the same cell type. Now, let's not forget that these ontology terms are the terms that we use, especially with disease, tissue, and cell type. They're mapped to an ontology, and the ontology itself has its own hierarchy of how the terms are linked to each other, right? So for cell types, we use cell type ontology. For disease, we use Mondo and disease ontology, tissues, Uberon, and for everything else, we use experimental factors. And our system also allows us to manage and maintain multiple versions of ontologies. So just in case, because as the ontology itself changes, sometimes terms get deleted or renamed or remapped to a different name. And we want to make sure that data sets that were curated before, we still capture and represent them in the graph so that we don't lose that association from when it was curated to the term it was curated to. And having a graph itself now helps us do a lot more interesting things on it. For example, if a scientist comes in or an analyst comes in and they want to find all data sets that contain myeloid cells, we go to the graph, we find the term, the myeloid cells, and then we traverse the graph using graph traversal methods, and we find all the sub-cell types of cell types that are children of myeloid cells, and then go back and find the data sets that contain those terms and then return that. So that's one sort of functionality that you can provide through the graph-based approach. And finally, the indexing process runs every night, at least internally, so that we take all the new and updated data sets from ArtifactDB, and then we index them in our graph. And we've been doing this for all of the RNA-seq and ATAC-seq data sets. And we only replicate the minimal information that we need to support these ontology-based searches, because for everything else, we fall back to ArtifactDB for providing the full metadata. So for anyone that is interested in technically, we use Neo4j as the underlying graph database. So the entire ontology itself is represented as a graph, and we track the relationships between different terms and data sets. For ontology itself, as I mentioned earlier, we use experimental factors, Uberon, disease, and other ontologies as well. And we maintain several versions of them. On top of the graph itself, we have a GraphQL API that is very flexible in what you want to query from the graph, and then that thing is hosted on an Amazon EC2 instance. This is also in the process of being open source, the entire underlying system that provides these graph-based searches. There's a Python API that lets you search the API and find data sets based on ontologies terms or any keywords. So for example, if you're doing any of these searches to find myeloid cells, you're not only finding data sets tagged to myeloid cells, but also to any of these subtypes. And you can also use the Python API to, in general, resolve ontology identifiers. You want to find terms by synonyms or by description, and so on. And the system is called Kokoro, and we're in the process of making it open source. So how does the system work in practice, right? So you would go to SingleCellHub, which is our internal application where a lot of our scientists go to, and there's a drop-down field to find cell types, data sets by cell type of interest. Once you type in myeloid cells, that then performs all the underlying search and then presents you with a table of all the data sets that contain myeloid cells. And once you click on a data set of interest, we have a very similar representation to an MAE. So MAEs are data sets, and within each MAE, you have individual experiments. So you can also look at each of the experiments itself, and one of the labels that you see for cell type is a myeloid cell. And we also provide quick visualizations to look at the reduced dimensions, for example, TSNE or UMAPs that are available for this data set. And you see that there's a label myeloid cell that is this yellow cluster on the plot. In addition, we also link internally to a lot of other applications that let you either analyze this data set or explore this experiment through a lot of the downstream applications. So Kana is also one of the applications that is open source, and also something that Aaron talked about in his previous presentation that lets you do a full single cell analysis in the browser. We also have an internal instance of that is connected to our internal data systems. So you can explore, you can explore the data set, but if you want to reanalyze the data set, you can go to Kana and reanalyze that as well. We also provide easy to use snippets to access this data set or experiment in various computational environments. So if you want to access this experiment in R or Python, we provide easy to use snippets as well. And in addition, we also support alternative experiments because that's something the single cell experiment object supports. So if this data set or this experiment contains alternate experiments, we also list them on the application. And any reference that was available for the data set is being shown on the application. For example, if the data set is coming from a publication, we provide a link to the publication, or if it's coming from Geo or any other sources, we provide links as well on the app. And as I mentioned earlier, we provide a few quick visualizations on the portal itself to explore these data sets. So on SingleCellHub itself, if you're looking at an RNA-seq data set, we provide a dimensionality embedding plot that lets you choose various embeddings that are available in this data set. You can color the cells by any of the annotations that are part of the call data of the object, and then also overlay the expression of a gene on this data set. The visual itself is also custom built by us. It's based on a GPU-based rendering library called epibase.gl. It's been engineered to render around 1 million cells in the browser without too much effort. And something that you can explore is one of the cell by gene data sets, the Tabula Sapiens, that has around 1 million cells, and it renders pretty quickly as long as the data comes back. And we also do similar things for accessibility data sets. So for single cell ATAC-seq data sets, you can visualize the ATAC-seq signal as tracks. This should be very familiar for folks who use the genome browser. So it works very similar. You choose a gene of interest. We then compute the pseudo-Welk accessibility signal for annotation that the user wants to, and then compute these scores and then overlay this data as tracks. This is also using one of our previous library called epibase for visualizing genome-like tracks. And finally, Kana is one of the applications that I mentioned earlier. It lets you interactively explore single-cell data sets. So in addition to those quick visualizations that we provide on the portal itself, Kana can also compute markers for each of the clusters that we identify in the application. So you can compute the markers for any annotation and then look at them. Or we also have a versus mode where you think, these two, the pink and the green clusters, are not really two separate clusters, but you want to identify what are the markers that actually separate those two clusters. The versus mode lets you do so. And then version 3, it's actually out soon, rather than what the slide says, where you can also compute feature set scores. So these are gene set scores, if you're interested from either Go or MSigDB. Something we're working on next is extending Kana to multimodal data sets. So if you have RNA-seq and accessibility data sets for the same cells, then you can continue to explore them in Kana. You can also do a full analysis of single-cell data sets through Kana as well. And Kana is an application that uses some of the newer technology called WebAssembly. And the idea for WebAssembly is you have underlying C++ code that you can compile it to assembly that the browser can understand, and it can provide the same performance as it can run natively. So far, Kana supports both site-seq and per-tip-seq data sets. It can perform data integration and batch correction. You can reanalyze subsets of cells, but in general, you can go through the standardized single-cell pipeline and then do the full analysis, all the way from QC to UMAP and t-SNE generation. And with that, I'm going to pass it on to Hector for cell state query.
All right, thanks, Jay. I wonder if there's questions we can take now before we move on to the next section, or. All right. No, that's fine. All right, so Jay talked about how we set up our infrastructure for essentially querying by metadata, these kind of curated attributes of the data sets. But as I mentioned at the beginning of the talk, our intent is to really understand how we use these data sets or the kind of data collections to understand cell state. And starting from the beginning, we're thinking about querying as one of the fundamental operations we want to support. So then we think, all right, well, how do we start thinking about query in terms of cell state representations themselves? And one of the things that we've started working on is this idea of finding data sets by the specific expression of certain gene signatures or programs. And I'll use as an example regulation of amyloid beta clearance, a gene ontology gene set, but the principal use case that is being used, where this is being used, is a scientist performs an experiment where he compares, say, in a mouse model, a particular treated versus untreated sample, identifies cell types or sets of cells that distinguish treated versus untreated, identifies a set of genes that might characterize that difference, and then wants to understand if they've seen that signature of genes that says something about the effect of treatment elsewhere. And that could be in other mouse data sets or by understanding some, mapping some genes also on the human counterpart. So can you find this kind of specific gene signature in other data sets? But for the purposes of the illustration, I'll just use this kind of amyloid beta clearance pathway. And I'll demonstrate it with a particular experiment as we go along. This is a data set in our collection. It's a survey of prefrontal cortex development. And we know specifically in this particular case that amyloid beta clearance, microglia play a role by, they eat pieces of A beta plaques. Astrocytes are involved in clearing that into the vascular system. So we know microglia and astrocytes are definitely involved in the process of clearing A beta. So if we do things appropriately, we should be able to identify this data set as a data set that contains cells where this positive regulation of amyloid beta clearance is ongoing. So what would that mean? We know, for example, TREM2 is a gene that's involved in this process. So if we, as Jay showed, we can go to the hub and ask, well, what does expression of TREM2 look like in this data set? And we'll find it there over in that cluster where microglia tend to be. But of course, TREM2 is not the only data set gene involved here. LARP1, APOE are other genes that are involved in this clearance process. And we can look at expression of those two and see how that looks like within this particular data set. So now, but the question is, what we really want is to find data sets that contain sets of cells that we think the expression of this signature is active. So the way we think about this is to do a few things. One, we want to define a scalar function per cell that is defined by this query. So we're not pre-computing any of this. Well, we're pre-computing some things, but once you give us a gene list or a gene signature, we don't want to pre-compute everything for, say, every gene ontology gene set. And as I said, the most important use case is experimentally defined gene sets. So we really don't have access to do this. So we cannot really pre-compute most of this. So based on the set of genes that we have a query, we want to define a scalar function. Because of the properties of these data sets and just because it's, statistical hygiene, I suppose, we don't really want to make inference based on the function of the value of this function in a single cell. We really want to think about cell sets. So we want to discover cell sets enriched for these high values of this function. And then at that point, think about controlling discovery rate and then returning those enriched cell sets. So I'll walk you through our initial instance of how we instantiated this general process. And remember, we're kind of trying to target the real-time query use case here. So we tried to do all of this quickly. And certain decisions that were made to allow that. So a decision that was made to allow that is, well, how do we define this scalar function? So we want to find places where we think this gene is expressed at a higher level than some background model. So, we taking a cue from things like what Seurat does or what ScanPy does. We build a cell gene specific background expression distribution, and then compute a cell gene specific C-score that captures expression of a particular gene in a particular cell relative to some background model. And once we have that as our scalar function, we then discover cell sets enriched for these high function values. So for a given cell set, we can look at the distribution of these C-scores and then decide if we observe enrichment for high values of this function. And again, for the purpose of trying to answer real-time queries, we go with a simple gene set enrichment analysis framework. Here, instead of gene set enrichment, we're looking for cell set enrichment. But we use a similar statistical procedure to define for a given cell set, do we observe sufficiently high values of this function? And part of the reason for the use of the Z-scores is because statistically, this turned to, it's not right, but at least it's meant to work with this particular enrichment approach. And at that point, we have per cell set inference. We have a set of p-values. We can do false discovery control at that point, and then find those data sets in which those cell sets are enriched. Right. And if we do that, that's exactly what we do. So you enter either a gene set, you enter a list of genes, and just like if you had entered a particular cell type label, we will give you data sets. And I'll give you data sets where we think it contains a cell set enriched for expression of that signature. And at that point, you can do everything that Jay just showed. But instead of searching by ontology and metadata, we're searching by status of the transcriptome based on this gene signature score. So as I mentioned, this is what you'll see. You'll see the same applications. You can do everything that you can, Jay showed you. But there was a little bit of an issue in what I just showed you. I said for statistical purposes, we want to make statements about cell sets, which is the right thing to do. Everything that I showed you, the cell set that we were using are cell types, which is not as useful because then you can just type cell type names and be done. All right. So we kind of want to think a little bit better about how we structure this. We want cell sets. We don't only want to use cell types, but this is work that we've been pursuing now to think about how we do this. So abstractly, the problem what we're thinking about is the following, right? So we have a scalar function per cell. This was gene signature score. These cells live in some space, and I just here a drawing of it. We have predefined cell sets. These are cell type label annotations. And what we're doing is finding those sets where we see enrichment for that scalar function. And as I just showed you, or in the process that I just showed you, we would be able to identify sets like this one, right? Where essentially a good amount of the population of cells in that set have a significantly or sufficiently high signature score. It would, however, miss cases like this where we might have subsets of those cell sets where we have significant or high value of that signature score. And you can imagine how this might be the case, right? So if one of the labels is astrocytes and all astrocytes are doing their thing, clearing out pieces of A beta, great. The other set might be microglia, and there might be microglia that work really, really well in certain contexts to do their phagocytotic job. There might be microglia that are not working as well, say in a disease context. And we'd like to understand for specific signatures, where do we see activity? And it may not be of all microglia, but rather subsets of microglia or substates of microglia. So then how do we identify these cases where this a priori defined cell type label does not allow us to find lower resolution clusters or sets of cells that might have enrichment? So this is work that we did with one of our interns who was at Berkeley, and we thought about graph enrichment rather than cell set or cell type enrichment. And here we borrowed some ideas from Milo, work in Marioni and Sarah Teichmann's work lab, where they were doing differential abundance testing on the k-nearest neighbor graph. Instead, we're doing function enrichment, or essentially signal enrichment in the k-nearest neighbor graph. Here they predefine neighborhoods based on a sampling procedure. We do the same. We predefine neighborhoods to cover the graph using a sampling procedure, and based on that, define those cell sets and find places where the signal is high. So as opposed to using cell type labels, we cover the graph with these kind of randomly generated cell neighborhoods and use the same process as before. The other way of doing it without predefining cell neighborhoods is to let the signal itself define the neighborhoods in which we're going to do that. So we start with the k-nearest neighbor graph. We don't define neighborhoods ahead of time as we did with inspired by Milo, but rather let each cell in the k-nearest neighbor graph take a value from this signal function or this enrichment function, and then define neighborhoods based on that. We also use a little bit of smoothing since these values are a little bit noisy, and to find appropriately sized sets. There's a parameter to control the degree of smoothing, but essentially we're looking for bumps in the graph that define high values, and we do that by identifying connected components after we threshold the smooth signal value, the z-scores in this particular case. And once we have these sets, do the same enrichment procedure to find those cell sets. All right, so with that, we've gone from finding our enrichment of signature scores in a priori defined cell type labels. We're using graph enrichment to kind of solve the more cell state-like problem where we might have enrichment at lower resolution than what was annotated at the cell type label. And this has its trade-offs, of course. So with that, I'll add one more way in which we're kind of using this cell state querying approach. Here, before we were given a set of genes, can we find sets of cells that are expressing that signature at a high level? The other extreme is rather than I take a subset of cells, just give me a cell transcriptome completely, and I find other cells in our data set that have a similar transcriptome. And for that, actually, Graham will be talking to you in July and presenting this work. So I'll just give you a little bit of a teaser. Using a neural network for encoding transcriptomes, which is trained in a very specific way using cell type annotations, using triplet sampling. Graham has defined a kind of 128-dimensional space where we think similarity has some useful meaning. And with that, once a scientist finds a set of cells or a cell in an experiment that they think might be interesting and is asking the question, where else or what other context might we have seen this cell, they can embed that through the trained network, find other cells that are similar in the feature space that was defined by this network, and carry out query that way. And that's exactly what we have in our system as well. Scientists can define a set of cells of interest, and we can query every other data set based on the transcriptomes of those cells and do exactly the same thing that we've been doing. So instead of typing an attribute name or providing a list of genes, you can provide a transcriptome and then find queries or find data sets of interest in that particular way. And then again, do exactly the same thing that we've been doing for now. Now, those are two kinds of what we call cell state querying, and Jay will tell us a little bit about how we've engineered those two systems.
All right, so for the signature score calculation, what we do is we do a little bit of pre-processing because we can pre-compute all the z-scores for experiment, and we store them as Parquet files with columns as genes. This is kind of different from the orientation that most folks are familiar with if you're coming from the bioconnector world, because rows are genes in that sense. But the reason we use this particular layout and file format is because when the analyst enters a gene set that they're interested in, we can quickly eliminate experiments that does not contain that set without having to read the entire file. We can just look at the metadata and say, all right, if even one of the genes is not from the set is not present in this file, we don't have to open the experiment and do all the post-processing or run the methods on that experiment at all. So we have a process that runs every night that computes the z-scores for all the new and updated data sets. And one of the things is, so we, as Hector mentioned earlier, we have a total collection of around 130 million cells, but that's across 2,000 data sets, roughly around 1,000 to 1,100 of them are human data sets, and the rest are mouse and another species. So when someone performs a gene signature search, that they're doing it by species. So in real time, we're looking at around 1,000 data sets and looking at the Z-scores and then following the rest of the process that Hector described in the methods. So the entire system is deployed in our AWS through a Fargate cluster, and we can perform a signature search on all the data sets in around 3 minutes. And I'll talk about a little bit about how we quickly distribute these tasks across a lot of the workers in the cluster. One thing to note is that we currently limit our auto scaling to around 100 workers. But that's not the limit of Fargate itself. That's a limit that we set ourselves in our deployments. But certainly you can increase the number of workers that run a lot of these pre-processing and Z-score calculation steps, so that your time to do a signature search on all data sets could be less than 3 minutes, depending on the time. And so the deployment model that we use in our both for signature search and similarity calculation is we have an API endpoint where the user submits a request or the application submit a request. And we use a fire and forget mechanism for this API that then goes into a cluster that then does a lot of the running the methods itself. And once the method is fully finished, the results get stored in a cache, and then you have to pull for when the result is available. And then finally the result gets back to the user or the application. And that entire process can be demonstrated through the SingleCellHub portal itself, or we also have a Python API where you can just programmatically send a request to the API and then get the results back. Now, the similarity also runs a similar deployment model, except that the workers here are now running a neural network model, and you can reuse this architecture to perform real-time queries based on statistical model inferences. So it's the same deployment, but using it for a different use case. As I mentioned, all of our applications and systems are accessible and integrated with computational environments. So you can programmatically perform most of the things that you do on Shub on our SingleCell portal through Python, not just querying for data sets and looking through for data sets, but also performing a lot of these analytical operations. With that said, I'm going to pass it on to Hector for what's next.
All right. So if you'll grant me 5 minutes, we'll stop. So as I mentioned, I think that a key idea that we want to pursue is I think we've done a good job in kind of identifying data sets, but that's super helpful for many of our tasks. But I think it's also kind of useful to start thinking about how, across data sets, what can we say about particular cell state. So I showed you a very simple representation of state that we can reason about. Is a specific gene expressed, or is there a signature that's expressed? And now we can start asking these questions about these very simple kinds of questions across data sets, not just in a particular data set, but to summarize say the expression of a particular gene in a particular tissue or disease context across the data sets that we find. So is ACE2 expressed in which tissue? Usually long, but what disease or what does expression look at across different diseases and so on? Or TREM2, is it expressed in what disease states as well? So because for the metadata querying, we have kind of this ontology side of things, we're looking into ways of summarizing expression across this entire data set, so we can start asking these questions about specific genes and gene pairs. Now we call this the Shub cube. Cube here is a callback to 2000s, 1990s data mining things where we're talking about data cubes and so on. We have ontology, so we have this hierarchical organization. We want to start asking these questions. Given a gene, find those tissues and cell types with highest median expression, and we can do that across specific ontology terms, and we can compute things across the ontology in real time. We also want to kind of define these things for gene pairs. I think that's a useful kinds of query as well. This is kind of our first attempt at kind of summarizing cell states across data sets, but it's also a reflection that once we rolled out our signature search that I showed you about, by far the most common signatures we were getting were single genes. So we have to do this soon. And then the other thing is that I showed you kind of, through similarity and neural networks and so on, kind of representations that do relatively well at identifying these similarities. I didn't show you that it works well. Graham will show you that in July, but it seems to work well for finding similar full transcriptome cell states. But it's kind of hard to understand exactly what's going on. So a lot of our effort is going into this question of how do we bridge this gap between representations that are easy to reason about, like gene signatures, with its peculiarities, and then kind of neural net representations with their own peculiarities. So things that we're thinking about here, and things that would be fun to think with others about this, is can we start thinking about network-based representations of regulations or TF activity, right? So we look for signatures, and that's kind of a catch-all for gene sets are a catch-all for different kinds of things. And ideally, we'd like to think a little bit more specifically, pathways. Maybe you want to represent in a particular way. The sets of targets of a transcription factor, you might want to think about it in a separate way. And in fact, the state of those targets, or how much activity of a particular transcriptome factor, you might want to think about it in a different way. So we might want to find data sets where we think a particular transcription factor is active. Just constructing a set of its targets may not be the right thing to do given our current state. So we want to think about specifically better ways of addressing those specific instances. We showed you similarity as a deep neural network way of thinking about this, but again, it's kind of a little disconnected from understanding signatures programs that might be interesting. So ways of aligning representation learning to known signatures of programs might be useful here. On a similar vein, kind of using attribution methods on these learned representations to query signatures and programs or subsets or specific gene lists might be something that's interesting. And something that I'm really curious to understand is how do we start thinking about representations and querying for co-occurrence, cell state co-occurrences in a cell population. So that's, for example, if we want to ask questions about, again, A-beta clearing, is there a particular subset of microglia that we tend to see with a particular subset of astrocytes that might be interesting to look at in certain contexts? And if we start answering these questions in methodologically, then how do we engineer querying data sets based on these representations? And I mentioned that it would be extremely useful not just to understand cell state, but also understand cell state organization in tissue. And that's where a lot of kind of spatial modalities would be useful. And obviously, interesting to start thinking about kind of similar questions in that space, which we haven't really done yet, but it's something we're starting to think about. And that includes questions about structural harmonization and how do we represent data sets, semantic harmonization, and what about tissue or spatial locations can be harmonized. And then the same questions about finding representations of cell state in their spatial organization, and then how do we query those? I think that's something that would be super interesting to think about as well. So with that, I'll leave you to it. There's a lot of work that's been gone into this and a lot of collaborations, a lot of great people working on this. I'd like to acknowledge and thank them and take any additional questions or comments in these few minutes that we have left.
Oh, great. Thanks, Hector and Jay. That was a pretty impressive run through an awful lot of work that you guys have done. And I guess we're open for a question or two. Does anybody from the audience have a question that they would like to pose? Well, I might ask you one, maybe even a different one than the last one I asked you about it, but how do you think about, in some sense, moving into a world where you have a little bit more data on a set of people, right? So it's nice to say, oh, let's find the difference between a non-diseased kidney and a diseased kidney. But one of the real big problems that comes from single cell data, I think, is that people think because I have thousands of cells from one person and these really sort of impressive averages and lots of data. We just don't have a way to incorporate the sort of between human variation yet in these models. And is that gonna sort of be a bridge too far in the sense of, like, if you start representing a bunch of interesting genotypes that you think might be associated with disease states, now, you know, you just have too much data? Or how have you guys thought about that?
Yeah, no, I think that is by far I think that's really where we'll spend the next, as a field, I think, in general, is kind of, you know, kind of transiting these levels of organization, right? I think really been thinking of cell population, and that's insufficient. I think with spatial we can start thinking about tissue, but ultimately what we really care about, you know, for any of our programs to succeed is how to understand patient level heterogeneity in these contexts. I think, you know, there's going to be ways in which people are going to do this, and we might think about those, right? We can start thinking, you know, I've been kind of pushing this whole representation, but it's representations of cells within a particular space. I think we can start, you know, wondering about how we're going to represent a full cell population that corresponds to the sample of one individual in some space, and place them in potentially some disease trajectory, and then ask questions about properties of those patients and how those properties map in a disease trajectory that's defined by kind of these molecular features, right? That for sure, honestly, we're starting to ask the question is actually what, you know, we've focused so much on cell population that we really haven't thought about what is the patient population, or the subject population, organism population that we have right now. And that's something we're starting to extract and starting to understand. We're in the process of just putting together a very, very, very big table, long table of understanding just what we have. And of course, it's going to be complete bias in how it was surveyed and how it was collected, but at least starting to understand how we start thinking about this would be useful. The other more direct way is on, as you mentioned, right? So for a smaller subset of samples in which there are genotypes and there are some data available between those, and we can make more direct connection between genetic variation and some of what we see in the phenotype. You know, I think that's data we haven't really touched much upon it. There's another kind of internal project that has been working on it. But I think the big open question is, how do you bring these two things together, right? So direct observation of the effect of genetic variability, kind of inferred variability that you observe or you construct from molecular profiles, and how do you then bring those two together in a way that allows you to say something about, you know, the full connection between genetic variation and what we observe as a disease trajectory. I think a combination of well-defined populations in which we can do the more direct mapping between genetic variation and the effect of those genetic variants, plus, let's say, less refined ways of defining those molecular states so that we can just observe a lot of it, so that we can kind of build those disease trajectories. The combination of those two, I think, will get us somewhere. And for sure, we've been kind of investing on the let's map out the space part of it by collecting a lot of data, but that in conjunction with kind of more better defined populations from which to define those genetic component, genetic variant effect component, I think would be useful. Ultimately, I think if we're thinking about the prediction side, I think a combination of both will be necessary, something to map out the space in which to understand the effect of perturbations, and well-defined either populations on the genetic side or perturbation experiments on the in vitro side will kind of get us a little bit closer. So there's never enough data, just blindly generating data. I don't know about that.
Right, thank you. And I think we're a couple of minutes over now, so folks, join in thanking Hector and Jay again, and we'll call the session to a close at that point.